Scientists have discovered a mechanism, or at least one mechanism, by which the measles infection can make patients susceptible to other diseases. Welcome to Microbial Minutes. This is ASM's update on what's hot in the microbial sciences, the Measles and Immune Amnesia edition. I'm Julie Wolf, Science Communication Specialist here at ASM, and today we'll be talking about a paper recently published in the journal Science that shows how the measles infection can make patients susceptible to additional microbial pathogens besides measles after they've been infected. Now, we know that measles is a very important human pathogen. This virus infects more than 7 million people annually and leads to 100,000 deaths each year. Reduced vaccination rates have led to a 300% increase in measles infection since 2018. And in fact, the United Kingdom has recently lost its measles-free status because of a fall in vaccination rates. And the United States was at great risk of losing its status as well. Now, this same group, whose research we are highlighting today, previously has shown that measles epidemics can be linked to population mortality even 2 to 3 years after the measles epidemic is over. And this research is starting to investigate the reasons why. In order to do this study, the researchers recruited 77 healthy but unvaccinated children who were in between the ages of 4 and 17. They took blood samples of these children before and after a 2013 measles outbreak, and they characterized the immune responses within the blood of these volunteers. One measure of immunity is to look at the circulating antibodies, which are molecules that can recognize various regions of microbial proteins. So the region of a protein that is recognized by an antibody is called an epitope. Now, a healthy individual will have not only many different antibodies that will recognize all of the different proteins on the outside of a pathogen, but a healthy individual should also have multiple antibodies that will recognize different epitopes of each protein. So as we look at their first test on the next slide, we can see that they wanted to evaluate how many epitopes were recognized before and then after the measles epidemic by the same individuals. Antibodies were tested against a variety of human viral pathogens to see how well they recognized different epitopes from these pathogens. And one individual was tracked before and after. If you zoom way in, there are gray lines together an individual's response before and after. The time points there are linked by a black bar, and that black bar is the average for the group. There are many different controls that are important to consider here. The A, B, and C are, as shown here, are different control cohorts for the unvaccinated cohort. In these cohorts, we have age-matched controls, which were either sampled at the same time in A or at a different time in B as the age-matched controls. And in control C, those are adults to evaluate what the adult immune response or the adult antibody repertoire looked like when tested at the same time as the test cohorts. The test cohort itself was put into those individuals who did not experience measles during this epidemic. They have no vaccination history and they did not experience the disease. Those are in green here. And then those that experienced either mild or severe measles disease, and those are put into the red categories on the far right side there, with the blue category there representing individuals who had been vaccinated before the study had begun, and then their antibodies could also be tracked throughout the course of the epidemic. What they're looking at here are the number of different epitopes recognized by the entire repertoire of the antibodies that are circulating in blood. And you can see most of those controls have a linear, a straight relationship. There is no change in the number of types of recognizing molecules that those antibodies can bind to. However, both the mild and the severe decrease the number of epitopes that the antibodies are able to recognize. And in fact, 12 of the 77 children lost over 40% of their antibody repertoire. Now, in the box area there, that's looking at just the entire repertoire of antibodies. If you want to look at some of the individual viruses and the responses to those viruses and how those were affected by measles infection, those are shown in the bottom, where the scientists broke out the enterovirus, rhinovirus, coronavirus, etc. And you can see very similar patterns as a decrease in repertoire is found mostly in the patients that experience severe measles and to a slightly lesser extent in the patients that experience mild measles disease as well. Now, humans have a whole lot of different variables that are harder to control for. And so to demonstrate that this loss in repertoire was connected to measles infection, the scientists conducted an experimental model of measles in rhesus macaques. We'll show you that on the next page. They tested 4 different animals, which are shown here. The animals are labeled with the number and the letter at the top. And the repertoire of antibodies are shown relative to the beginning before they were experimentally infected. Each monkey lost on average 40 to 60% of its pre-existing antibody repertoire. And so this, combined with the patient study, really emphasizes the measles virus as one that can affect not only the immunity to measles, but immunity to other potential pathogens, either those that you have experienced in the past or those that you may experience again in the future. On the next slide, we'll see that this was picked up in a number of different areas. This is a very important finding, so it was broadcast in a number of mainstream media, including The Guardian, who highlighted not only this particular study, but another companion study that looked at the measles virus ability to wipe out some of the immune cells, not only the effector molecules, but the cell populations themselves. This had a very nice schematic which demonstrates that measles can decrease both the memory component, so those infections or antigens or microbial proteins that you may have seen previously, and the naive cells, so basically the potential repertoire of what an individual may be able to react to should that particular antigen or microbial protein be encountered. That was highlighted in addition to this antibody study showing that the effector molecules of some of these populations all in tandem are leading to a decrease in immunity within measles patients. The lead author of this study, Michael Mina, was quoted within an NPR story where he said that the real takeaway is that underneath the surface, measles is much more than a rash, and it's much more than just an acute viral infection. It's got these very long-term, stealth-like detrimental effects that are extraordinarily difficult to measure. So today we've heard how measles virus can diminish pre-existing antibodies that offer pathogen from other pathogens. I'd like to thank you for listening and thank Ray Ortega for production. If you want to hear more updates about measles virus and other things related to infectious disease and immunity, be sure to subscribe and ring the bell so that you get notifications when we have a new update. I'm Julie Wolf, and I'll be with you next time on Microbial Minutes.